I'm not seeing who is in the, here we go. There we go. Hello, everyone. Good afternoon, and welcome to today's webinar, Unraveling Immune Exhaustion Genes and Racial Disparities in Early-Onset Breast Cancer using the All of Us Dataset, hosted by Community Campus Partnerships for Health and RTI International. Before we begin, please feel free to use the chat to share questions, comments, reflections, and emojis throughout the session. Let's begin by sharing the institution or organization you represent. Next slide, please. And I'm not seeing the slide deck. There we go. And you can stop. Thank you very, very much. And we can continue on. I want to make sure that everyone feels welcome. And I will continue with the agenda. Of course, we've welcomed and done the logistics. We're going to talk about a little bit about Community Campus Partnerships for Health. Then Dr. Sula Hood will do a presentation on the Researcher Academy, and Dr. Oyomari Osazua Peters will present. We will then have Q&A, and Dr. Owai, who is Dr. Oyomari Osazua Peters, will do a Q&A with Dr. Tomi Ekenyamiju, also from Duke University. Once the Q&A and discussion is complete, we will have announcements and updates, and we will have attendee insights from CCPH and closing remarks. The evaluation survey, our attendee insights, is an opportunity for us to hear from you. Your comments and insights are important to us, so please take a few moments at the end of the webinar and fill out the survey. Thank you in advance. Next slide. Community Campus Partnerships for Health promotes health equity and social justice through partnerships between communities and academic institutions. We view health broadly as physical, mental, emotional, social, and spiritual well-being, and we emphasize partnership approaches to health that focus on changing the conditions and environments in which people live, work, study, pray, and play. Next slide, please. And here is our Community Campus Partnerships for Health, All of Us Researcher Academy team. You can see our names. I would just like to highlight our Executive Director, Al Richmond. Next slide, please. And now you will hear about the Researcher Academy from Dr. Sula Hood. Dr. Hood is a senior research scientist at RTI, a nonprofit research institute. She has over 17 years of experience with a focus on health communication and community engagement in genomics. Dr. Hood is a co-investigator on RTI's All of Us Engagement and Retention Innovator Award, and she serves as a co-lead for the All of Us Researcher Academy. I'd like to just remind you all to please share in the chat any questions you may have for Dr. Hood, and she will respond during the Q&A. Dr. Hood?
Hello. Thank you so much for that great introduction. I'm going to be sharing a little bit about our All of Us Researcher Academy today, as well as talking about the All of Us Researcher Workbench. But before I get started, I just want to acknowledge my team. As you can see, we have a large team that works very hard and in close collaboration with CCPH to make these types of events and webinars and offerings possible. So I just want to acknowledge my team. There's way too many of us to go through individually. Next slide, please. All right. So I'm going to talk a little bit, as I mentioned, about what the All of Us Researcher Academy is. But first, I would like to talk about what All of Us is, especially for those of you all who might not be familiar with All of Us. So the All of Us Research Program is a NIH-sponsored effort, and it's a historic effort to collect data from 1 million or more people across the United States with an emphasis on informing transforming precision medicine and advancing precision medicine and individualized care. And so there's a lot of information and data that are collected from individuals that are available for analysis by researchers. And so where we fit in terms of the All of Us Researcher Academy is that we provide training and technical assistance for researchers who wish to analyze the All of Us data. The way data is analyzed within All of Us is through the Researcher Workbench, which is a cloud-based platform. In addition to making connections for researchers to be able to access the All of Us data, we also provide, as I mentioned, training and technical assistance on a variety of different research-related topics that are relevant from analysis to dissemination. But we also consider ourselves to be a learning network, and we have many activities that are in service of fostering peer-to-peer support and fostering connectedness among researchers. Next slide, please. All right. So we have a variety of different types of offerings through the Researcher Academy, and I'm going to share about a few of those with you all. First, I'd like to talk about our Institutional Champion Awards. And these are awards, funding awards of up to $75,000 that are awarded to institutions for teams of researchers to work together to analyze data. These are teams of researchers from across different disciplines, and they also go to support faculty members, their students, or other trainees such as postdocs. To date, we have provided institutional champion awards to 20 institutions. We're very proud of that across 4 different groups, as you can see. And our reach that we've had for these awards have spanned across the Eastern and Western US. Next slide, please. Okay. As I mentioned, part of a big component actually of our Researcher Academy is a focus on training. And so we have curated over the past few years a wide range of course offerings. These are short courses, and we have made them available on demand. And so they are freely accessible to individuals that would like to visit those course topics. We do have a Academy course catalog available. We have the QR code here that you can scan if you have your phones out, or we will have a live link in the presentation deck, and you can see a description of each of our courses. But they range, as I mentioned, from introductions into All of Us to data analysis, or how to write up publications, how to incorporate the All of Us activities into your courses if you are a faculty member, for example. So a wide variety of different types of skills. Next slide, please. All right. Another aspect of our Researcher Academy, as I mentioned, is the building of a learning network. And so we try to have a wide reach. And one of the ways in which we do that is through our Academy newsletter. This is a newsletter that comes out on a quarterly basis. To date, we have almost 800 subscribers. And this is how individuals who are connected to our Researcher Academy can learn about all of the great things that we offer, whether it's these types of webinars that we're doing in partnership with CCPH, or if there's a specific course, or we also do promotion for some of our partners as well. One of the other things that we like to do is to spotlight and highlight the research of our Academy members, such as our interns or our institutional champion awardees. And so it's an opportunity to support them and enhance their visibility. I highly encourage you, if you are not already a subscriber of the newsletter, you can scan the QR code here, and it's a quick and easy process to get on our subscriber list. Next slide. All right. So now I'm going to talk in the next few minutes about the All of Us Researcher Workbench. As I mentioned, individuals who are interested in analyzing All of Us data use this virtual platform to be able to analyze the data. Next slide, please, Angela. Okay, thank you. So as I mentioned, the Researcher Workbench is a cloud-based platform which houses the All of Us data, and it is very secure. If you look over here to the right, there's all different types of data, which lends itself to be able to be analyzed by researchers from various different types of disciplines. There's hundreds of thousands of survey responses on behavioral data, environmental data, all different types of data. There's physical measurements data, wearables data from Fitbits. There's also genetics and genomics data available as well for researchers who are interested in those analyses. When individuals are looking to work within the researcher workbench and sign up for the researcher workbench, I do want to make you aware that there are two tiers of data. Our speaker for today will talk a little bit more about which tier that she used, but there's first the registered tier, and this level or access level of access data access contains individual level data for approved researchers who have registered for the workbench. And so data such as health, electronic health records or wearables or survey data. However, the control tier of the researcher workbench is where our more sensitive data is going to be available, and that includes genomics data as well as some of our health records data that might have suppressed demographic fields that were not accessible in the registered tier. Next slide, please. All right. So, in order for individuals who are not registered users of the researcher workbench to access the data, their organizations that they are affiliated with will need to have what's called a data use and registration agreement, or a DURA, in place. And so this is a organizational kind of sign off or affiliation. And then once an individual goes to sign up at the individual level, the DURA will enable them to be able to be linked as a person who can actually access the data. To learn more about the DURA process, or to even see if your institution may possibly have a DURA in place, you can scan this QR code, and then you can actually see the list of institutions that have one. Okay, next slide, please. And there's a bit of an echo. Okay. All right. So now I'm just going to briefly walk you through the process of actually signing up for the researcher workbench. Step one, once you have confirmed that your institution does have a DURA in place, then you go through the process of creating your account. There is a mandatory ethics training around using the All of Us data that you do have to go through. And then step four is to sign the data user code of conduct. And there's something on the screen there, Angela, if we can remove that. But we do have a QR code here on the slide, which will take you directly to the registration. Next slide, please. All right. So once you get to the point of setting up your account, this is very important here. There will be a question that asks you, how do you learn about the researcher workbench? If you are learning about it through us, we would love to be able to have some credit for connecting you to this great resource. And so you would select activity or presentation or event. And then for the next question, and it asks, did you learn about the workbench from any of these program partners? And we've circled here that the All of Us Researcher Academy / RTI International is on the list. And so you can click on that. Next slide, please. All right. So without further ado, I would like to connect you all, and um, I would like to connect with you all uh, with our next speaker. I know you're going to be doing an introduction as well, Angela. I'm just really excited. So I've told you about this great resource in the researcher workbench, and so now you'll be able to hear about an actual researcher who's been able to leverage this resource. So thank you again. I'll be able to take any questions that you have in the chat, or feel free to reach out to me. Thank you.
Thank you, Dr. Hood,
Mhm.
for that comprehensive explanation about the Researcher Academy and the workbench. Right now, I'd like to introduce everyone to Dr. Oyomare Osazuwa Peters, our second presenter. Dr. Peters is an assistant professor, or as she's also known as Dr. OY, is an assistant professor in the Department of Population Health Science, Duke University School of Medicine, with a primary research interest in characterizing the interplay between social factors and biological characteristics as drivers of poor health outcomes. She is a member of the Regal team at Duke, which actively pursues and promotes research to eliminate global cancer disparities research. Dr. Oy, we're glad to have you.
Thank you, Angela. Thanks for that introduction, and thanks for the opportunity to present our work. So I am delighted, to be telling you about this project on immune exhaustion genes as risk factors in early onset breast cancer. And I'll start by telling you a little bit more about me. I have a background in biostatistics and genetic epidemiology, and I've spent quite a while using traditional statistics to evaluate risk factors for a wide range of outcomes. However, in January 2024, I had the opportunity to be part of Emma Head's training program using the All of Us dataset to learn to apply machine learning approaches on big data. So the project that I'm going to be presenting, the research study, is actually a product from this training that I went through with Emma Head using All of Us data. So for people listening to us, we're glad that you're here. We want you to understand how researchers like myself are making use of the All of Us data set to uncover patterns and trends that promote equity in healthcare outcomes. So here's an outline of how the talk will go. I'll start off by giving some background, defining the research questions, showing you how we made use of All of Us database, going through our questions, the methods, and what we found, and then bringing it all together by summarizing and stating what the future holds for this research question. So let's start out with some background. Like it's commonly known, breast cancer is the most commonly diagnosed cancer amongst females globally across the world. Sadly, some populations do more poorly than others. And on this slide, we're giving an example. African American women are more likely to die from breast cancer than white women. And the pattern is consistent. So on the slide, you are seeing results from two different studies, one using the SEER database and one using NCDB database. And on the x-axis is showing you the hazard ratio, which is a measure of the risk of death for one group relative to the other. So here we're looking at African American women compared to white women. Notice that, as indicated by that red box, African American women have a 71% higher risk of death compared to white women due to breast cancer. That's for the SEER database. And then when we look at NCDB, it's a 22% higher risk of death. So however you look at it, it's consistent. There's evidence that some populations do more poorly than others. The question is why? So then we turn to incidence rates for aggressive breast cancer. It appears that African American women tend to have the highest rates of very aggressive forms of breast cancer. So if you look at the figure A on the left panel, the x-axis is showing you incidence rates, age-adjusted incidence rates per 100,000, and the So that's the y-axis. The x-axis is showing you year from 2010 to 2019. And what you see, the blue line that's above all of the other lines is showing the trend, temporal trend for African American women. And you can see on average, it's way higher, even more than double the rates for other populations. So the question, though, is how then does this contribute to the disparities we see for breast cancer? Emerging evidence suggests that a biological phenomenon called immune exhaustion might play a role. So what is immune exhaustion? Well, it's an attribute of immune cells. They are supposed to mount an attack on infection or tumor cells. However, when immune cells become hyperresponsive and are not able to carry out their usual function, this is described as immune exhaustion. So to illustrate, let's look at the figure to the right using a T cell as an example. So whereas immune exhaustion can happen across different immune cells, it's been most studied focusing on T cells. So if we start out with a naive T cell that's surveilling the body and then is exposed to antigens or proteins from tumor cells or from infection, the usual process would be for that naive T cell to transition to an effector T cell. So the blue T cell, the blue cell right there. And then on your right, the next step after the effector T cell fights the infection or fights the tumor cell and eliminates it, it then becomes a memory T cell. However, when you have continued infection or continued antigen persistence, as you have with cancer, it is possible for that T cell to go through the process on the left, where it becomes exhausted. There's a lot of inflammation. There's an upregulation of multiple inhibitory receptors like LAG3, CTLA4. At the end of the day, the immune cell becomes exhausted and it's not able to mount cytotoxic activity to fight the tumor cells. It's interesting that there are consequences when you have high levels of exhaustion. So again, looking at this figure to the right, when you have high levels of exhaustion, it is associated with poor prognosis for patients with cancer. Whereas if you look to the left, when you have low levels of exhaustion, then you can predict a higher chance of good prognosis. So let's come back to breast cancer though. The patterns that have been shown in the literature is that immune exhaustion tends to be more prevalent amongst individuals of African ancestry. So what you're seeing here on the x-axis is showing you the median difference between white and black individuals. When the bars are red, it indicates that you have higher levels amongst black individuals. When the bars are blue, it means you have highest levels amongst white individuals. And on the y-axis, you have markers of immune exhaustion. Notice that most of the bars are red, both in the TCGA study cohort and the WCHS study cohort. So that tells us that immune exhaustion is more prevalent amongst individuals of African ancestry. So now what does this mean? Well, the evidence from the literature shows that genes that regulate immune exhaustion process are well known. However, there is less known about the contribution of these genes in the risk of aggressiveness. But the patterns we see in the literature suggest there might be a connection. So this knowledge gap sort of defined our research question. We asked, are immune exhaustion related genes associated with aggressive breast cancer? And then how does this association vary by race? Now take note that in this study, race is a proxy for social disadvantage, because again, there's evidence that suggests that immune exhaustion, there's an association between the stress people experience and the levels of immune exhaustion. So here we use race as a proxy for populations that experience different levels of social disadvantage. So that takes us to our data source. We have a research question. What data can we use to answer that question? So we turn to the All of Us database. At the time, we had access to version 7. And as Dr. Hood has shown us, all of us has different kinds of data. But to answer our question, we needed electronic health records and genomic data. So that implies that we needed access to the control tier, meaning that like Dr. Hood mentioned, you have more sensitive data. It means it has more protections to make sure it's secure. And so there are 4 requirements that I went through to be able to access the data. First of all, I established that Duke University has a DURA with the All of Us program. I created an account and verified my identity. I went through the mandatory trainings required to access the control tier, and then I signed the data user code of conduct. By the time I was done with all of those requirements, I had access to the researcher workbench, which is a cloud-based platform that gives you access to the data, but also provides you the analytical tools that you need to work with the data. So as I described, the methods I went through, you can imagine that all of this was completed in the researcher workbench. So that takes us next to the question we asked and how we defined our study sample. So first of all, we identified all of us participants that had a diagnosis of malignant breast cancer, but then we applied filters. We required that they also had condition start dates. We really needed to know when the breast cancer was diagnosed. We required that it was a primary malignant breast cancer. So for individuals for whom it was a secondary diagnosis, we had to exclude them. We also required that all of the individuals were unrelated. So if there were related participants, we had to exclude some of them. And so our final sample size was 2,896 individuals who were eligible to be included in our study cohort. Now that takes us to the genetic variant. So we have our study cohort, and now we identified all of the genetic variants in the All of Us database, over 48 million of them. We restricted that number to genetic variants that were within 785 genes that have been identified as being involved in immune exhaustion through the nanostrings immune exhaustion panel. And then genetic variants can be classified as either being common variants or rare variants, depending on the minor allele frequency. So for common variants in the population, we applied standard filters and ended up with about 126,000 of them. For rare variants, when we applied the standard filters, we had over 394,000 of them. For outcome, we were interested in triple negative breast cancer subtype, which is one of the most aggressive forms of breast cancer, but that information wasn't available in the database. So instead, we used early onset breast cancer because TNBC, triple negative breast cancer, tends to occur at early ages. So we defined early onset breast cancer as a breast cancer diagnosis that occurred between 18 and 50 years. As you can see in the graph, for those who had early onset breast cancer, there was a slightly more frequent proportion amongst women who were Black or African American. So 26% of them had early onset breast cancer, whereas among white women, it was about 22%. So now that takes us to answering the questions. For common variants, we use candidate gene analysis to filter, because we had a lot of variants. We wanted to identify the ones that are actually associated with the outcome. So we use the candidate gene analysis, which basically tests for the association between the genetic variant and the outcome while adjusting for race and genetic ancestry. And the goal was to only select those that had statistical significance and then move them into a second phase of analysis where we use machine learning models. So here you are looking at the result from the candidate gene analysis. This is a Manhattan plot. On the x-axis, it tells you where the genetic variant is located in the genome, what chromosome. And on the y-axis, you have a log transform p-value. And the dashed line shows the threshold for significant. We had about 66 variants meet that threshold. However, to move to the next phase, they had to have non-missing information, meaning no NAs, because machine learning models do not do well with NAs. So only 48 of these variants met that requirement, and we moved those into elastic net regression model. And using machine learning approaches, we identified the best model. And the best model had an area under the curve of about 0.68. And it identified 11 genetic variants that were significantly associated with our outcome. It also showed us that being Black was associated with a 23% higher odds of having early onset breast cancer. Although that odds ratio was not significant, but we could see the evidence of the pattern. Now, elastic net regression makes an assumption that the relationship between the outcome and the predictor is linear, but we know there are genetic variants that might have nonlinear associations with the outcome. So we considered another random forest model, another model called random forest, which can account for those nonlinear relationships. And then this model identifies the top 10 predictors of the outcome. Amongst the top 10 genetic variants associated with early onset breast cancer, we identified 4 additional genes that were not captured by elastic net regression. So you can think of this model as being complementary to what we have shown before. Now we've talked about common variants. We also have rare variants. Do we find evidence of an association with aggressive breast cancer? To deal with rare variants, the typical statistical methods do not work. So we have to use a different approach called rare variant Bodin association test. So what happens is you deal with the rare variants on the gene level. For each gene, and looking at this illustration, we have 3 individuals in the cohort. In the particular gene, we're looking at the sequence. For each gene, we group the rare variants. We score each individual by summing the rare variant count per gene. So for each individual, how many of these rare variants do they have? And that becomes a new genotype, a new score. So rather than dealing with each genetic variant, we're sort of putting that information into a new variable, and then we are putting that variable in a model and testing for the association between the variable and the outcome using linear regression. So we do that, and we found 1 gene that was significantly associated with our outcome. You're looking at a violin plot with the log transform p-values on the y-axis and the beta coefficient estimate on the x-axis. And GATA-A3-AS1 did show significant association with early onset breast cancer. So that answers question 1, takes us to question 2. To answer question 2, we basically repeat the same models, but this time separately for non-Hispanic Black only and separately for non-Hispanic White only. So what do we find? Again, we started out with elastic net regression, so you are seeing a similar table to what you saw before, but now column 3 is showing the results for the full cohort, column 4 shows the results for Black only, and column 5 shows the results for white only. So notice that for Black only, 4 genetic variants were significantly associated with the outcome, while for non-Hispanic whites, there were 9 genetic variants that were significantly associated with the outcome. 9 of those variants were fully captured when we looked at the full cohort, but for the Black group, 2 out of the 4 cohorts were not captured in the full cohort analysis. So that tells us there was more overlap between Black and white. Okay, similar pattern when we looked at random forest. You have more overlap with the white group and the full cohort compared to the Black and the full cohort. So let's put it all together. We asked the question, are immune exhaustion-related genes associated with aggressive breast cancer? And we found evidence that some genes show association using the different machine learning models and the rare variant analysis. When we looked at it by race, we saw there was an association, although non-significant. And then we also saw from the different models we looked at that the specific genes associated with the outcome were different depending on if you were looking at black or white. So when we did some functional annotation, thanks to contributions from a postdoc in our lab, we saw that whereas for black people, the genes were involved in immune suppression, but that function was not captured for non-Hispanic whites. So here we're picking up subtle differences. There were some limitations in carrying out the study that we need to acknowledge. The sample size for Black was really small. We did not consider other racial and ethnic groups. There were a few challenges working with the All of Us researcher workbench, but I must mention they have a great help desk. They have office hours. So if you're facing any challenges, you can really get help. And then you might ask, why are we studying all of this? Well, it has potential value for precision medicine because studies like this can inform designing population-based specific screening, like what is done for Jewish women. So the slides went off, but essentially, we think that the work we're doing has good value for making precision medicine available to all. So at this point, I want to say thank you to my team, contributors, Dr. Akinyemi Ju, the postdoc, and my mentor in the Emma Head program. And I think at this point, we can invite Tommy to join the conversation. So Tommy is the principal investigator for VEGAL, the research team at Duke that studies, that does research to eliminate global cancer disparities. She's a professor and she's an expert in cancer and social epidemiology, focusing on modifiable risk factors that underlie disparities. So Tommy, welcome.
Yeah, thanks. So great presentation, very impressive findings, and I think are really important for cancer prevention and treatment, as you said, and really helps us, one, describe for others what a valuable resource the All of Us dataset is, and also to discuss some of the novel insights that we've been able to generate using that data in a way that can help shed light on this persistent issue of breast cancer disparities that we've been talking about for many decades, and unfortunately haven't really seen much progress in terms of closing the gap. So really appreciate your analysis. And I think, yeah, we would like to invite anyone that has questions to put those in the chat for OI to address. But while we do that, was there anything about the results that surprised you or that you found counterintuitive?
I think not so much the results, but walking through the whole process, I was surprised at how much work has been put into the All of Us program. When you're on the outside, you really don't have a sense of what a massive investment it is. So that I thought was surprising, intimately learning about how much people are involved, even learning about the ROTI folks who are involved in this academic research program. That was all surprising to me as an outsider. But the other thing that was counterintuitive was the fact that despite all of these efforts, we still have small sample sizes for non-white groups. So it tells us that we have a long way to go and we have to put in more effort, to increasing the representation of the diversity that is in the US population.
Yeah, that's a great point. And to your point about the All of Us being a really valuable resource, it's a cohort, right? It's a cohort that includes all individuals, all disease types. So it's not specifically focused on cancer. And so by the time you restrict your cohort to looking at cancer patients, and then you restrict it to breast cancer patients, and you're trying to restrict it even further to triple negative, it gets smaller and smaller, right? So it's just a testament to the amount of time and effort and funding and resources that's needed to generate this large cohort in order to be able, in order to enable people like us that are interested in very specific diseases to be able to even have any chance of making generating meaningful insights. And as you know, in our lab, we also have studies ongoing where we're recruiting patients, where we're trying to collect survey data, collect biospecimen. We know very intimately that this is very, very hard work.
Yeah.
It is extremely hard work. So just kudos to the All of Us team and all the work that you all do in the background. And even just cleaning the data is by itself a massive endeavor, so that by the time somebody like OY gets to it, we're ready to do the analysis and go from there. So how about in terms of the results that you found, some genes that were only identified for Black patients, and then that the findings for white patients was similar to what we found in the overall cohort, but not true for non-Hispanic Black patients?
Yeah, and I think I didn't get to show Jovita's picture, but the postdoc in our group did a great job of functionally annotating these genetic variants that we found for the different groups. I think it was striking and to pick up on that subtle difference. Even though there were 9 variants for non-Hispanic white and only 4 variants for non-Hispanic Black, non-Hispanic Black seem to include a wide range of functions. Those variants for non-Hispanic Black included immune suppression, right? But we didn't find that function for non-Hispanic white. So it's the gut feeling I have is like we're onto something here. We might need larger powered studies to get the exact variants that matter, but the evidence we're getting is there are differences that need to be described and not ignored.
Dr. Owai, I'd like to just interject because there's a question in the chat.
Okay.
It's asking, based on your work, can you recommend health education outreach strategies that involve social determinants of health?
So I'm going to take a stab at that question while Tommy also prepares her answer. And I think for me, the profound message from my work is getting all communities to be involved in participating in a program like All of Us. Because if we do not participate, so talking about social determinants of health, we want a representation of people across board. Because if we cannot study them, we cannot provide the information that's needed to inform effective interventions. So we do not want only some people, we want all sorts of people across the spectrum, across the social determinants of health, including access and all that range. We want to be able to capture that diversity. So the message for education and outreach is to get as more people as possible, as diverse as possible, to participate in the All of Us program so that they are represented and captured in the data. Tommy, what do you think?
Yeah, I think that's very important. There's the practical consideration, meaning, even being able to participate in a study like All of Us requires some level of investment of time and resources and raising awareness, which we know are domains of social determinants of health. So participating in a study like this, might be a bit more challenging for people who have less resources to start with. So raising awareness is key, but also paying attention to how we message about the study and how we, make it easy for people that may be less well-resourced to be able to participate is also key. And I think also thinking broadly about social determinants of health involves, if we play this out a few steps down the road, our goal is that these findings and understanding what is driving at the molecular level disparities in aggressive breast cancer, so that we can understand that mechanism, design, clinical trials, and develop targets that are able to address that mechanism for Black patients, for example. thinking of social determinants of health, we also have to make sure that at the end of the day, when we do have that drug and we do have those therapies, that people are able to access it, they're able to get the best quality treatment available. So it's a big question. There's a lot of work that needs to be done. We need to get our communities engaged and aware. We need to do provide tangible support to address some of the social determinants of health that prevent people from participating in research and being able to access the best quality care. And I think we just need more work like this to showcase what it means scientifically, right? If we're successful in doing that.
Thank you, Dr. Akinyemiju. There is another question that was placed in the chat. And it says, thank you, very interesting talk. I might have missed that information. Was the SNP genotyping data germline or coming from tumors? If it was tumor data, were you able to control for germline variation associated with shared genetic ancestry? Is there a possibility that some of these polymorphisms linked to exhaustion show differential allele frequencies between individuals of African and European ancestry? I know that was a multi-layered question. If you need for me to repeat it again after you begin answering, I am happy to do so. Thank you both.
No, I can see the question on this end. Thank you, Kyriaki. So first of all, the genomic data in all of us is germline. They collect blood samples and then sequence the genome. So it's definitely germline. And your question is great. And that I think I missed that slide. Our research group, we have designed a project called MEND, where we are going to actually dive into the tumor and examine differences in immune exhaustion biomarkers amongst white, African American Black, and West African Black. So we do have a study that's sort of addressing the question you're asking, but for the All of Us database, it's purely germline. And the findings from our study provided useful information to inform our design of the study that will look at tumor differences. In terms of accounting for allele frequencies, we had genetic ancestry PCAs, principal component axes. We had them in the model to account for those differences. But great question. Thank you. I think there's another one. Do you see the other question in the chat, Angela?
I do. As long as you see it, feel free to repeat it and answer.
Okay. So Sazul asked if we ever checked for a particular gene for SNP disparity. We didn't take that approach, Sazul. Our approach was kind of agnostic at the beginning, where we conducted the candidate gene analysis. We took the approach of what genetic variants are associated with the outcome in the study sample. So the gene you are pointing out, I don't know it, but I figure there might be something you know about the gene. Maybe it's been implicated in some in cancer outcomes, but that would be a different study design. In this particular case, we were interested in genes related to immune exhaustion, and we used NanoString's exhaustion panel as a way of identifying those genes. But when we subset to those 785 genes, we took an agnostic approach and allowed the data speak for itself. Basically, we tried to identify variants that were associated with the outcome. So a different approach from what you're suggesting, but the future opens up opportunities to consider different ways of finding answers to the question. Thank you. I don't see any other question.
Dr. Owai, can you talk a little bit more about the limits and challenges and to precision medicine and the way that your research can affect precision medicine? Because I think that's an interesting potential future for the research that you're doing.
Right, great question. So I think I also missed the slide where I mentioned population-based informed screening. So for example, Jewish women at the age of 25, they get screened for the BRCA mutation based on studies that have shown that this mutation occurs at a high frequency in this population. Well, studies like mine and maybe larger size studies that can give us, specific and definite information, they are key and fundamental to extending the benefits of precision medicine to other groups, because you cannot design such screening without knowing, what mutation a group, a particular population is more prone to have. So you need foundational studies that provide the information. And that's where studies like mine are important for extending the benefits of precision medicine to different groups. Tommy, do you have any thoughts?
I think that's a great response. The other way to think about, the exposure we're looking at in this case, immune exhaustion, tying it back to the point about social determinants of health. We know that immune exhaustion is not just a consequence of, chronic infection, chronic inflammation over time, and maybe pathogen exposure that happens repeatedly, making the immune system, exhausted and unable to cope. But we also understand that the social environment that we live in can also create stressors that through activation of the HPA axis can lead to, again, systemic inflammation, right? So all of those repeated daily stressors due to a lot of social conditions that people live in can also contribute. So that's another potential mechanism, linking social determinants of health with immune exhaustion. So thinking about it that way, it also makes a lot of sense that we see that non-Hispanic Black patients are more likely to have aggressive tumors, and that there's a genetic component to that. So really opens up a lot of new avenues for research. And the goal is that ultimately, if we can find a particular mechanism or a particular gene, that really raises the possibility that it's something that can be targeted with medication. Ideally, we'll be doing a lot of work further upstream to reduce stresses, to reduce the exposure in the first place. But once somebody has been diagnosed with breast cancer, if we have been able to prove definitively across many studies where the results are validated that immune exhaustion is a mechanism that drives tumor aggressiveness, then we can make the case that targeted therapy focused on that particular mechanism or gene at the time of diagnosis can really make a difference in improving prognosis for patients. And if we do that, make sure that patients have access to the highest quality care, including any genetic testing that may be required to be eligible for treatment, then we can really make progress in reducing disparities, making sure that all patients have the best possibility of survival after diagnosis.
Thank you, Dr. Akinyeju. I have another question, Dr. Orwai. If you could go into a little bit more depth about the MEND project that you mentioned, because it seems to be providing a broader data set, looking at a broader group of people of African ancestry.
So MEND is Tommy's baby. I'm going to pass it back to her because she can give us very good details and paint a very clear picture, much better than I would.
So, if my kids hear you say that, they're going to fight with me tonight. Like, no, we're your babies. So, but it was my first baby. Yeah, I actually, so this is a comprehensive study of breast cancer. And like I said, our goal is to be able to study women of West African descent, because as we know, due to the transatlantic slave trade that brought West Africans to the Americas and other parts of the world, there's a shared genetic background and ancestry there. When we look at triple negative breast cancer, we actually see one of the highest global prevalences in West African women. So there seems to be something specific to that population that may be driving the distribution of this aggressive subtype. We also see that in the US, African American women are significantly more likely to have triple negative breast cancer, and their rates are closer to West African women than to women of other populations. So MEND is really designed to help us explore this, as we've mentioned a few times. There's not a lot of data that have granular level information down to subtype data for diverse enough populations to really be able to answer this question rigorously. So our goal, we now have about 1,200 West African women that were diagnosed with breast cancer. About 40% of them have triple negative subtype, which is again, one of the highest global rates that we've seen. We're now recruiting African American women in the US and white women in the US to compare the distribution of those subtypes and to, really, delve deep into some of those mechanisms. So we have data on social determinants of health, access to care. We have blood, we have saliva samples, we have tumor tissue, we have normal tissue from patients. So this will really be a great resource to complement what the All of Us data set has, because it allows us to go into a lot more detail, significant sample size, and study a wider range of ancestral backgrounds, because we can look all the way from West Africa to European American patients and understand what those genetic differences might be. So more to come on that. We are actively working on this, and we'll be happy to share the results as they become available.
Dr. Kinyemiju, thank you very much for that in-depth explanation. We are excited and interested to follow your research and to hear the results. Dr. Owai, thank you very much for a very, very engaging presentation on early onset breast cancer and gene exhaustion. Dr. Hood, thank you very much for always giving insightful, in-depth information about the Researcher Academy and the Researcher Workbench. Thank you all very much. Keisha, next slide. As we continue to wrap up our time together, I want to draw your attention to the next webinar from the All of Us series, which will be on June 12th at 4:30 p.m., at which time we will be exploring health together, what the All of Us data set can teach us. You can register here right now for this webinar. You will also be sent information about this webinar. But want you to be able to put this on your calendar now. The focus around exploring health together will be on mental health. So please feel free to join us on June 12th at 4:30. Next slide, please. And now, again, I'm going to ask you all to please take a few moments, click on the link or use the QR code to share your insights on this webinar. We need and we want to hear from you. If you can just take a few moments, this helps to shape our future work. So thank you for taking the time for attending both this webinar and for taking a few moments to fill out the attendee insight. It is very much appreciated. All submissions are read and we use it to continue to further our work and to make it more engaging and interesting and relevant to you. Next slide, please. Just thank you. We can go back to the insight, the attendee insight slide. So that others can have time to fill it out. But I do want to thank all of you for attending. I want to thank, of course, our presenters and speakers. Want to thank you for your questions and look forward to starting a new conversation with our June 12th webinar on how the All of Us data set we can all learn from that data set. Thank you very, very much. Again, just take 10 more seconds to fill out that insight. And we appreciate all of your attendance here, as well as our speakers. Thank you all very much. Have a wonderful rest of your afternoon and enjoy the upcoming Memorial Day weekend. Thank you so much.